Okay. So what we're going to do today then is talk about this paper. There we go. Oh. People are doing a lot of this kind of stuff. But I picked this paper because it talks about an environment we're a little bit familiar with, and because I happen to know some of the sites they took it at, and Phil Hugenholz is a colleague. Filamentous chloroflexi are abundant in wastewater treatment processes with biological nutrient removal. This doesn't really tell you what the paper is about so much. Most filamentous bacteria, excuse me, in biological nutrient removal process, they're talking about wastewater treatment here. Processes have not been identified beyond morphotype and simple staining reactions. This is a truism. If you go to the Cary wastewater treatment plant, they've got this book, and it's just got filaments of different shapes and colors and things, and you stain them with things like, acridine orange or whatever, and it just uses a basic identification of these organisms, not by name, but just type 2 this or type 4 filaments or whatever. The majority of sludge filaments observed under the microscope do not hybridize the commonly used phylogenetic probes for proteobacteria, actinobacteria, firmicutes, etc., because they aren't any of those. Specific CNS ribosomal targeting all of those probes were designed in this paper for chloroflexi and optimized for use in fluorescent in situ hybridization. These samples are actually probably the worst case scenario for fluorescent in situ hybridization. The hardest part about fluorescent in situ hybridization isn't the actual procedure, it's something called background fluorescence or autofluorescence. What if your sample is already fluorescent before you do the staining? Now, why would a sample be fluorescent on its own? Can you guys think of anything that might be fluorescent in nature automatically?
Certain bacteria just already have fluorescent proteins.
Well, yeah, particularly photosynthetic ones, right? What photosynthetic pigments are fluorescent. And so anything photosynthetic is going to glow, and you have to do something about it. There's a bunch of stuff. I mean, this is wastewater sludge. There's all kinds of stuff in there. And so fluorescence is a problem. So they're used to doing the worst case scenario. Now, realistically, what you usually do is you put it under the microscope and you turn your laser up to 11 and you scan the whole thing before you stain it to bleach all that fluorescence as best you can. But some things you just can't bleach. Let's see. Chloroflexi have been implicated in sludge systems by culture independent method, blah, 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 blah. Predominant morphotypes responding to the probes were filamentous. In other words, most of the chloroflexi that they were seeing are filamentous. This is no surprise because most of the chloroflexi that we know of are filamentous. And were generally abundant in 10 Australian full-scale and 2 laboratory-scale sludge samples examined. Filamentous bacteria were subdivision 1 were rare, but 3 were common. We'll talk about the rest of this later. It is suggested that filamentous bacteria belonging to the chloroflexi account for a large fraction of phylogenetically uncharacterized filaments in wastewater systems and are likely to be abundant in such systems on a global scale. That's certainly true. Here's the thing. It is generally agreed by people who do wastewater treatment that filamentous bacteria in wastewater sludge is a problem, that they cause foaming and bulking. And so remember what we're thinking about here. Wastewater treatment facility, the raw sewage or raw effluent, whatever it might be, goes through some kind of steel gratings to get rid of big stuff, alligators and guns and all that kind of stuff. You'd be surprised. And then it's pumped into this giant tank. And it sits there. You typically go through an anaerobic process where it's set there and it just rots. Then typically you turn on some stirrers to mix it up and you turn on aeration, bubbling, to add oxygen to it. At this point, you get an aerobic, it's supposed to be aerobic, you're essentially pumping oxygen into it and burning up as much stuff as you can into CO2 and methane, well, not so much methane, and nitrogen that can blow off the system. Then you pump it into a cesspool and let it sit anaerobically, and then you make a bunch of methane. But a critical stage in that is the separation of most of the liquid from the sludge. It's only the sludge that you pump into a cesspool. That process, after aerobic digestion, requires that you can turn off the bubblers and the stirrers, and all the bacteria in there flocculate and settle to the bottom. And it's a surprisingly efficient process. And you get a hard layer of sediment at the bottom, sludge, that you pump into the cesspool. And the bulk liquid, you just pump out, either treat it with ozone or UV light or something to sterilize it, and it goes directly into the rivers and streams or whatever, some distance measured upstream of where water is taken in by other processes. So it's got to be pretty good. Wastewater treatment absolutely depends on your ability to get a clean separation there, and where most of the liquid you can dump into the stream, and it's only a small volume that you have to treat as sludge. Bulking and foaming are caused by a failure of that process, where either the sludge doesn't settle down hard and it's a big thick layer that you have to deal with, or it actually floats to the surface, in which case you can't separate them at all. And it's generally agreed that it's filamentous bacteria that cause that problem. We'll see that that's not actually generally the case. So there's some talk here. Fluorescence in situ hybridization using 16S RNA targeting oligonucleotide probes is an invaluable technique for directly identifying microorganisms in their natural setting. So that's what we talked about that. When they came into this paper, only species-specific fish probes were known. There were no broad phylum-specific. Wouldn't it be nice if you could have a set of probes that you could use to hybridize, and anything that was a Chloroflexus would hybridize and would light up? People have done this for a long time. There are probes for planktomycetes, he mentions. There are probes that light up most bacteria, but not archaea or eukaryotes. There are some probes for proteobacteria, etc., firmicutes, of varying quality. But until this paper, there weren't any decent Chloroflexus probes. And so, the aim of the present study was to design phylum and subdivision, that is subphylum, specific oligonucleotide probes for Chloroflexi and to evaluate them in these sludge samples for use in fish. And then to prove that they're usable by determining the abundance, morphology, and spatial distribution of Chloroflexi in activated sludges. These are the samples, the environments they're looking at. These are all, with the exception of these are all wastewater treatment facilities. This one is an industrial wastewater treatment facility. These ones are all municipal wastewater treatments. These are also agricultural ones. This one needs to remove carbon only. It's an organic compound that is in this sludge. I think it's phenol, if I remember correctly. This is all municipal waste. These then are agricultural waste, and so they're rich in phosphorus. So this one has to remove carbon. These have to remove carbon and nitrogen. These ones have to remove carbon, nitrogen, and phosphorus. And then these are two lab scale fermenters, digesters, that are specifically designed to remove carbon and phosphorus or carbon and nitrogen. What they've done then is they've looked at filamentous organisms in these environments. And so you just take the flocks, you look at them, and these are scored subjectively. I hate this part of this paper. They look at them and they evaluate subjectively how abundant filaments are. There's a wastewater treatment scale that they use for this from 1 to 6, or 0 to 6, where 6 means that there's an overabundance of filaments, and 0, of course, means there aren't any visible at all. And all of these, at least all the municipal and real life ones, not so much this one lab scale one, have lots of filaments in them. I'll get back to these because these are based on the probes in just a minute. So to start with, of course, they have ribosomal sequences from Chloroflexus from the databases, but it's unclear to them whether those are representative of the ones that might be in this environment. And so they go in and extract DNA from a municipal wastewater sludge. I think that's the NUSA one. Amplify ribosomal RNA, clone them, sequence them. They got 6 clones that were Chloroflexus. Excuse me, these are from the laboratory scale ones. Sequence them and analyze them, and they're sure enough, they're Chloroflexus, and they're shown in the tree. Right here. So this is a tree of Chloroflexus, all different kinds. The ones that are dark are cultivated species, Chloroflexus aurantiacus, various strains, Chloroflexus aggregans. And the ones that are highlighted are these 6 that they identified in their process. You can't read that, but it's in the paper. They're going to use these as controls. And so they took all those data, all those sequences in that tree are ribosomal sequences that are available for this phylogenetic group. And then they used them to design some probes. So they've got this one, which is an oligonucleotide sequence that was used before as a PCR primer, specifically for Chloroflexus. And then they've got these 3 that they designed based on alignment of all these ribosomal sequences that I showed you briefly in that tree. And they designed them to be as universal as they could make them. And so they have CFX 1223, 109, and 754. And they target, these numbers are the part of the ribosomal RNA that they hybridize to. These are the sequences. Notice that they're pretty well defined. This one has an M there. That means amino. If I remember correctly, that means A and C. And so there's a 50-50 mixture of A and C in the oligo at that position. Length, calculated TM. By the way, people who calculate TMs are usually fooling themselves. Oligonucleotide probes, melting points are a lot more complicated than just GC content, which is what that usually is based on. And then optimal formamide concentration. It turns out that when you do a hybridization, you can adjust the stringency and specificity very nicely by increasing something, by putting in something that also likes to make hydrogen bonds with bases. And that something can be urea or it can be formamide. And so if you look at hybridizations, you very often see urea and formamide in there, either to disrupt base pairing or to increase the specificity. For each one of these probes, they have a positive control, something they know from the sequence ought to hybridize directly to it, and they like to use herpetocycline for this. And then a negative control, something they know from the sequence should definitely not hybridize to it, but they try to make it as close as they can. And so this number represents how many mismatches there are between this sequence and the ribosomal sequence of that organism. And so they're looking for things with a single mismatch wherever they can, because they want these to be really specific. Now in the case of this one, the 1223, the closest they could get was 2 mismatches. Now one thing I didn't mention was when they pulled through, they pulled the ribosomal sequences out of that sludge to get the 6 full-length sequences. They actually threw one of them away. And they threw it away because it was a chimera. This is in the book. I didn't talk about it in class. But when you amplify ribosomal RNAs from the environment, there's an artifact that can be created. PCR is a very powerful technique, and that means it creates powerful trouble sometimes. Imagine a DNA polymerase running down a ribosomal gene, and it hits a big hairpin loop in the RNA because the single-strand DNA wants to fold into that same structure, right? It's going to pause there until it can open up that helix, right? And so this means that when DNA polymerase amplifies from a ribosomal sequence, it does so in fits and starts. It doesn't run smoothly down the DNA from one end to the other. It stalls periodically along that sequence. Now what happens if it's never released from that stall? The next round of the PCR is a denaturation, and so it gets separated. And now you've got a partial length piece of DNA. That partial length piece of DNA, if it ends in a conserved sequence, can serve as a primer in the next round of amplification. It can anneal to a different sequence and serve as a primer, and it will be elongated from that. And now you have a sequence that's from one organism at one end and a different organism at the other. There are a variety of ways to deal with these. You always have to look for them in environmental PCRs. And the way they did it is the most basic of them, is that they took the 5' end of the sequence and the 3' end, and they did phylogenetic trees separately. And let's see, he actually talks about it, I guess, in the results. But this particular clone was a Chloroflexus at one end and something else at the other end. I think it was an actinobacterium at the other end. This sort of thing happens all the time. You always have to be on the lookout for them. They were probes were synthesized, and they were 5' labeled with fluorescein or with Psi3 or Psi5. These are 3 different colored dyes, fluorescent dyes. That is, that they emit, they fluoresce in 3 different colors: red, blue, and yellow, I think. Here's a cool trick. Because they have 3 different colors on 3 different probes, that means you can do the hybridizations all at once. And you can look for one color, another color, or more than one color mixed together. The blending of those 2 colors means that the organism is hybridizing to more than one probe. And that's information you can use to identify the organism. And so with 3 dyes, you actually end up with, what, 3, 4, 5, 6, 7 different color combinations that you can look for. That's pretty cool. And here's the subjective scoring method, which is awful. Here we go. One further clone, SBR 1064, proved to be chimeric. With a partial, you can actually, if you do this carefully, you can identify where the breakpoint is between the 2. At 12:15, the 5' fragment belonged to the Chloroflexus, 99% identical to this other clone. And the 3' fragment belonged to something else, to a candidate file. So something way off the beaten track.
So question.
Yeah.
Why did they say E. coli nucleotide? Like,
Oh,
if
so-
it wasn't from.
Yeah, so this is a nomenclature thing. I'll pull that back up again.
That just kind of threw me off.
Yeah. Oops. Approximate breakpoint at E. coli position 1215. Ribosomal RNAs, the nucleotide positions are numbered based on E. coli. And you make an alignment and you use the E. coli numbering so that when you say 1215, you know what nucleotide it is in the secondary structure of the RNA instead of, because these primers don't amplify the whole ribosomal RNA, so you don't know what number it is in the original sequence. And this 1215 is a specific place, it's a specific nucleotide, It's the same way the tRNAs are numbered from 1 to 78 because you want to look at homologous residues when you're doing this. Good question. So the next step is to evaluate them on pure cultures, to optimize the hybridization. Here's the key. As with all broad specificity probe, it was difficult to design probes that hit all sequences in the target group. That's an understatement of the paper. So here's the results of this. Now this is a prediction. This is not from their data. I'm going to zoom out as far as I can go on this because it doesn't matter what the organisms are. They have the ribosomal RNAs of all these sequences in this tree. And so for each one of their probes, the first one is the UB mix. This is a mixture of probes that should hybridize to all eubacteria. And these are then each of their other probes that they design. There's a black square here if that sequence should hybridize to that probe based on its sequence. So this is a prediction. You'll notice that even with the so-called universal probe, the eubacterial probe, there are organisms just in this group that aren't going to hybridize to it. This is why when you do your PCRs in lab, you don't always get a PCR product. Because if you happen to have this organism as a colony on a plate, it won't work. Yeah?
So they have highlighted one that did not hybridize to any of them.
Yeah.
How did they, I guess maybe I'm misunderstanding it, but how did they know that was present?
Well, it's not present. So these are sequences from a database. So these are all Chloroflexus sequences that were in the ribosomal database and were long enough at that time. This would be like for the term project, if those of you who started to look at this, if you were to click on the whole phylum Chloroflexi and make a tree. Notice that there are some that are gray. These gray ones are ones where the probe isn't included in the sequence. So these probes are somewhere along the sequence. And what if the sequence in the database of the ribosomal RNA is incomplete and doesn't have that region? And so they don't know whether it will probe or not. Or the sequence isn't high quality, and so they're not certain whether that probe will hybridize or not. There might be an N in the sequence, for example, that's in the database, and so they don't know for sure. And they include them here because this collection it's not the greatest collection of sequences in the world. Notice that they have some specificity. This probe, and I don't remember which one it is, it's supposed to be specific for this subclass, the Chloroflexus 3 subclass. And you can see it hits it pretty well, but there are a bunch of missing, but at least it doesn't hit things outside of the group. This probe is supposed to hit group 1. It hits group 1A pretty well, not so much group 1B. And so this is the difficulty with ribosomal probes and hitting different groups of organisms. It's easy if you want to target one species, because you have one sequence, you know what it is, you put it in a hypervariable region, so there aren't going to be anybody, it's not going to be anybody else that has it. But when you're trying to hit swaths of organisms, probe design is a nightmare, and you really have to put some effort into it. But these are the probes they end up with, and you can see what they hybridize to and what they don't, and how specific they are or aren't. Their recommendation is they take their two general Chloroflexus probes and mix them together, because when you combine those two columns, you get really good coverage of the Chloroflexi, and one of the things they don't show in that image is all the other bacteria, because just as much as you want to hit all the Chloroflexi, you don't want to hit anybody else, right? And so let's take a look at the pictures. These are flattened three-dimensional images. And so they've taken laser confocal microscopy, They take and slice it through a sludge flocks, individual flocks of this activated sludge, and then collapse the images so they can print them on a piece of paper. The blue is background luminescence. And so this is just background autofluorescence. And so you're looking for white in this case. These are hybridized. I have to look at the. A is Rose Hill, 2 probes with good overlap, and that's why they're responded to U mix. Okay, so I'm sorry. In this case, the blue is the eubacterial mix, and so that should hybridize to all the bacteria in here. Because it's hybridizing to the red and the yellow, I think it is, probe, you end up with a white filament. Because the organisms are hybridizing to both probes, and it's the mixture of color. Here you've got some red probes that are hybridizing alone. Look at these filaments. In some cases, they're everywhere. They're also able to identify organisms that hybridize to one probe and not the other. Let's see. So, yeah, okay, so it works. We demonstrate filaments representative of Chloroflexus are generally abundant in these activated sludge. One of the things they point out for themselves, I'm betting anything that a reviewer demanded this, is that digital image analysis methods are available for a definitive microbial quantification. They just numbered these 1 through 6, right? It turns out that there are methods where, particularly with 3-dimensional images, a computer can scan through those images and add up the length of all the filaments and use that to actually measure the density of filaments in the population. They needed to do that. Most of the sequences that when they did the molecular phylogenetic survey, when they fished sequences out of the environment, they got mostly Chloroflexus 1, subgroup 1. But when you use the probes, what you see is Chloroflexus, which ones? Fish, Chloroflexus, or more? 3. Yeah. Let's see that, but it must take
The beginning of the sentence.
Okay, got it. Okay, I'm sure. So 3. And so here's one of these cases where PCR bias or cloning of sequencing gives you a difference in quantitatively who's there. And that's why it's important to do this fluorescent in situ hybridization if you want to know how many are there. Non-quantitative nature of PCR. Herpetocycline had previously been found to be responsible for bulking. However, yeah, okay, so they think that these things are herpetocycline. There's a great, this is a great method to study wastewater sludge. Somewhere in here, it must be this bit that you can't see yet, is going to make you worry about it. The investigated sludges were not suffering from bulking. In other words, these were normal, properly functioning wastewater treatment plants. You saw those sludges, they're full of filaments. And some of them were up in the 5 and 6 range in terms of the number of filaments. And so and there's lots and lots of Chloroflexus in there. And so this makes you worry about the general common knowledge in the wastewater treatment world that these organisms are the cause of bulking and foaming. The reality is there's lots of them there, whether these treatment plants are functioning properly or not. There's one difference. When you look at these, and they looked at this in some detail, this is a sludge flock. All the filaments are inside it. You don't see filaments branching and connecting sludge flocks together, which is the danger. This is what causes specifically foaming, because then that traps air bubbles. Those networks of flocks trap air bubbles, which causes them to float. And so maybe chloroflexus isn't the problem after all, or maybe it's only certain kinds of chloroflexus that are the problem. And so a large part of what this paper does, other than the technological advancement, is that it brings into question this common knowledge that the appearance of lots of chloroflexus in sludge is a sign of trouble. That may very well not be the case. All right. So on Friday, we'll do the discussion session, the microbial handome, right? What do you know about the microbes on your hand? I'm going to be sending out an email describing some things about the so-called Think Project. How many of you are familiar with this on campus? Yeah, well, this class is part of that. I haven't told you that, but it is. And and it's particularly the discussion sessions. And so those of you who get emails from Think are likely to get emails. I see some frowns over here. Are likely to get some things from that, and you'll be asked to submit some information about the class. And again, it all has to do with the discussion sessions, and I'll send you an email with all that information. All right, thanks.